Oi oi, Mr. Wells here. This is monoclonal antibodies, which is a triple-only biology topic. Let's start with antibodies, which are these little Y-shaped molecules. They are made by a specific type of white blood cell called a lymphocyte. Antibodies are produced when we are infected by a certain disease, and they're specific to the disease, the pathogen that causes that disease. They bind onto it and attack it. Monoclonal antibodies, monoclone, suggests that it's made from a single clone, a single copied from one single cell. To produce monoclonal antibodies, we need to isolate a single lymphocyte that is producing the antibody that we want and then cause it to clone itself, to copy itself loads of times, so that we make an absolute buttload of that antibody that we're interested in. To achieve this, we stimulate a mouse's lymphocytes to produce the antibodies that we want by injecting, say, the pathogen into the mouse. Here's the lymphocyte we have isolated from the mouse, which is producing antibodies that are specific to the thing that we injected that mouse with. The problem with this lymphocyte is that it cannot multiply. So in order to make this cell divide so that we've got loads of factories producing loads and loads of antibodies that we're after, we add this to a tumour cell, a cell that's dividing rapidly. Lymphocyte plus tumour cell gives us this cell, which is called a hybridoma, the mix of those two together. So this is a rapidly dividing antigen-producing cell. That hybridoma that we produce divides, leading to the production of an absolute buttload of antibodies, all identical to each other. That is the reason we call this monoclonal antibodies, because they all originate from the same clone. Now, because we can produce antibodies that will seek out just about anything inside of our bodies, it gives these monoclonal antibodies a lot of uses. Monoclonal antibodies are used in pregnancy tests, so when somebody is pregnant, they produce a certain hormone. The monoclonal antibodies can seek out that hormone. We can use them in research, so we can attach a fluorescent dye to the antibody so that we can use it to sort of seek out and make it easy to identify certain molecules in cells or maybe different pathogens in the body. We could even use them to treat cancer. So we can attach a radioactive isotope to the antibody so that the antibody then takes that radioactive isotope straight to the cancerous cells and only damages and destroys them. Despite all these really exciting possible applications of monoclonal antibodies, they've not been as widely used as we first thought when this procedure was discovered. They produce way more side effects than first expected. They're also really expensive and time-consuming to produce. And in a sense, they're a bit too specific. You can only ever treat a single disease at a time with monoclonal antibodies.